Hello everyone, I'm Vivek Patni, and today my lecture will cover the selective methods for the bacterial growth. So today we are going to study the methods which are used for the bacterial growth. So why do we use this selective? Why the word selective? Because different microbes grow in different types of environments, and these environments basically helps us also to identify different types of bacteria through the selective methods. That's what selective methods is composed of. So starting with the lecture now. As you all know, a natural microbial populations basically in the environment, okay, it is very large and a complex one. It is very large and a complex one. Okay, large means very numbers of microbes are there, and a complex variety of different varieties of microbes, such as maybe fungi, it may be bacteria, it may be a protozoa, bacteriophages, numerous of them. So many different microbial species normally inhabit the various parts of our body also, such as oral cavity, intestinal tract, as well as skin. Okay, and as well as in the soil, in the tree, in the water bodies, etc. Numerous, there is numerous of examples, and these microbes are basically present in huge, extremely huge numbers. For example, a single sneeze, if you sneeze a single time, okay, it may disperse about 10 to power 3 to 10 to power 5 number of bacteria, meaning thousands to lakhs of bacteria in a single sneeze. And just a 1 gram of your feces may contain almost 10 to power 11 to 12 bacteria. Okay, so that is the number of microbes, not only numbers but also the diversity in the natural environments are also high. So another example, let's take from an environment in a soil, a fertile garden soil, okay, which may have billions of microbes per gram, and includes various species of microbes. It may be bacteria, it may be fungi, it may be algae, it may be protozoa, etc. Okay, so the study of the microorganisms in these habitats require knowledge of the specific microbes which are present in them. So in order to study this, we need to grow these populations. Okay, and basically we need to isolate them to study each one and them. If we need to study the mixed population also, there needs to be a specific selective method. Okay, that's all what we are going to study. It's just an overview only, just the basic methods which we are going to study now. Okay, the methods which are used basically, selective methods which we are going to study, as well as what are pure cultures and how do you maintain and preserve pure cultures, and cultural collection centers which are present in the world. Okay, I just listed a few of them. There are numerous number of cultural collection centers. Now starting with the selective methods. So the number of the organisms of the choice or the organism to be purified may be very less in the natural sample. It is possible. These selection methods can be broadly classified into 4 different categories. The first one deals with the chemical methods of the selection. Second one deals with the physical methods of the selection. Third one, the biological methods of the selection. And the fourth one is the selection in the nature itself. Okay, why do we need these methods? Its a reason is simple. Methods which we use may hinder the easy isolation and purification of organisms of our choice. So we need to be specific with our method which we are going to use a particular species of microbes. Okay, so we can overcome this problem by adopting the certain selection methods, which basically increases the chances of the microbes we want to isolate. Okay, by creating a set of specific conditions, which basically enhances the growth which we are trying to isolate. Okay, and also in some cases we can also inhibit the unwanted organisms or even kill the other organisms in the method. Okay, now if you want to view the complete video, please download the Science Edutech app, and the link is given in the description. I'm repeating it. If you want to view the complete video, please download the Science Edutech app and purchase the course Basics of Microbiology, where you can view the complete video. Thank you, and also subscribe my channel, and also subscribe Mr. Sagar Sonar's channel.